All right, well, thank you all for coming today and for sticking out till somewhat towards the end. I'm really excited to share our work with you. This is something that we are just finishing up, so it's not out yet. But what we study in our lab are many different things, but primarily what I'm interested in is the gut immune system and how it deals with the wide variety of different challenges that it faces. So the gut is an important barrier site, which interfaces with the outside of the body. And through this, that means that the gut can have exposure to a lot of different antigens, including food, bacteria, toxins, viruses, fungi, et cetera, all of which can either be pathogenic or harmful, beneficial, or anywhere in between, depending on the quantity and the context. So the gut really has this enormous task of trying to balance protection and homeostasis when faced with all these foreign antigens. And something that mucosal immunologists have to deal with all the time is how does the gut manage to do this? How does the immune system balance these two kind of competing tasks? And one way that it does this, although certainly not the only way that it does this, is through IgA, which is the predominant antibody isotype secreted into the intestinal lumen. So here's a little diagram showing various functions of IgA. It can neutralize toxins in the gut, coat different microbial components like bacteria, fungi, and viruses. We've shown from our lab previously that it also coats food antigens in both healthy humans and allergic humans. And then it can also exclude pathogens from accessing that mucosal barrier. And for a bunch of these reasons, this is the reason that IgA is kind of the goal of mucosal vaccination. If you can make a protective IgA response at this barrier tissue, it's much more effective at preventing initial infection and then therefore severe disease down the line than a circulating antibody would be after you have already been infected with an agent. So one question that has been really challenging for people to answer and that we've tried to tackle in kind of our own way is how is high affinity antigen-specific IgA generated in the gut? Now, I know we've heard a lot of talks in different model systems today, but I just wanted to start out by saying this, except for one experiment that I'll show you, is all in mice. We know a lot about antigen-specific B-cell activation in peripheral lymph nodes. So by peripheral here, I mean just non-mucosal draining. And we know that this requires things like antigen-dependent activation of B cells and T cells, TFH cell help and germinal center entry, affinity maturation of those B cells in the germinal center, and then plasma cell and memory B cell differentiation into these effector B cell kind of cell types. But does antibody induction in the gut follow the same rules? There's a lot of literature out there, but to kind of summarize, there's many reasons why we think that it doesn't. Namely that the structure of the lymphoid tissues in the gut is quite different than that in peripheral sites. Additionally, we have a piece of preliminary data from our lab that we've been hanging on to for a while and will hopefully get out there someday, suggesting that when you knock out TFH cells and therefore have no germinal centers in the gut, high affinity, that is NP2 specific IgA, is still intact, indicating that the germinal center is not necessary for antigen-specific and high-affinity antigen-specific IgA induction in mucosal tissues. So one question we wanted to answer was specifically, what is the role of the GC in IgA production? Now, to do that, we wanted to turn to a different model system. So instead of knocking out the germinal center and seeing if it's required, we wanted to observe B-cell participation in the germinal center and see how that contributes to the eventual antigen-specific plasma cell response. So using this GC fate mapping mouse, we can give mice tamoxifen, which induces expression of this S1PR2 CRE-ERT2. And when this CRE is expressed, you will get permanent TD tomato labeling of any S1PR2 positive cell that will continue throughout its lifetime and the lifetime of all of its descendants. Now, S1PR2 with very high fidelity marks GC B cells. So in this system, when we give tamoxifen, then we induce TD tomato labeling in GC B cells, and any cells that are derived from the germinal center will remain red. So we can actually then look at the eventual antigen-specific plasma cells, and based on whether or not they express TD tomato, we can say whether they were part of a germinal center response at one point in time. So looking at this, we used our typical oral immunization model where we provide oral ovalbumin and cholera toxin as our mucosal adjuvant 6 times once a week to our fate reporter mice, and we give them tamoxifen throughout the entire labeling window. And then we look at weeks 3 and week 6 to see the proportion of plasma cells that are OVA-specific and IgA positive in the lamina propria, which is where gut plasma cells home to, that are GC-derived. So here TD tomato equals GC-derived. And at week 3, we can see that some of them are, but most of them are actually not coming from the germinal center. Whereas at week 6, this is the opposite. We have the majority of the plasma cells that are antigen specific are from the GC, and a minority are actually non-GC derived. And this is quantified here from 3 different experiments. Now, this was very interesting to us. We really haven't seen this kind of temporal biphasic control of GC entry before. In regular sort of peripheral immune responses, you would expect the GC independent maturation to happen on the order of days, but here it's happening on the order of weeks. So that's definitely something we could investigate a little bit more. But we were also really curious as to how the GC-derived plasma cells at week 6 were different from the non-GC-derived plasma cells at week 3. So something that we did was we tried to kind of strike a balance between the week 3 time point when most of them were not from the GC and the week 6 time point when most of them were GC derived, and go to week 4. So we took down mice at this time point, collected gut induction sites like the mesenteric lymph node and Peyer's patches, and then also the lamina propria where the plasma cells home, sorted out antigen-specific B cells that were TD tomato negative and TD tomato positive, and submitted them for 10x single cell gene expression and BCR sequencing. And all the analysis for this and other experiments was done with a lot of help from the Kleinstein lab, including Hailong, Gisela, and Christine. I am the biology side of this. They're definitely more on the computational side, and their help has been extremely useful throughout this project. So what we were able to do is cluster the B cells we received into the TD tomato positive and TD tomato negative fractions. And what we can see, first of all, gratifyingly, is that there's not really any GC B cell population in the non-GC derived portion, which was a great sanity check. But we were able to define naive/memory, which clustered together B cells in both TD tomato positive and negative, as well as plasma cells. So to characterize these a little bit more, we first looked at isotypes and mutation burdens. So when we look at isotypes of plasma cells in the lamina propria, that are, actually, these are just all plasma cells from all 3 sites, we can see that they're primarily IgA, which is something that has been, very well described. And then also we've seen this by flow, but there are some isotype switched plasma cells that are not IgA, but this doesn't seem to really be different between GC and non-GC derived cells. Likewise, we don't see any difference in somatic mutations between cells from the GC or not from the GC at this particular time point, indicating that this non-GC pathway of plasma cell differentiation is still able to include classical B cell maturation processes that occur associated with or in the germinal center, like somatic hypermutation and class switching. In contrast to this, we were surprised to see that the memory B cells didn't follow that same trajectory. So really class switch memory in our system, although there wasn't a ton of it, does seem to come from primarily the germinal center, whereas this non-class switched memory/naive cluster is really enriched in the non-GC fraction. And then there's any evidence of somatic hypermutation is happening in the germinal center, not in this non-GC portion. So that really seems to differentiate the signals required for the development of kind of class-switched mutated IgA plasma cells from class-switched IgA memory B cells. We also wanted to look a little bit deeper at the mutations. As we all know, not all somatic mutations are created equal, particularly the ones that cause replacements in amino acid changes and that also occur in the CDR regions, which interface with antigen, are more important for the affinity maturation of the antibody. And we do see a significant, although small, decrease in the frequency of CDR replacement mutations here in the non-GC fraction, although there are still some. And when we quantify using Shazam and the baseline method, the selection pressure that is experienced by lamina propria IgA antigen-specific plasma cells from the GC versus the non-GC portions, we see that both of them are experiencing positive selection. We're not really powered to make any sort of claims about the difference between these 2 peaks, but it is interesting to us that similar to the CDR replacement mutations, they're both experiencing positive selection, but it appears at least that the non-GC is experiencing it slightly less so. So to summarize the first part, I've shown you that the germinal center increasingly contributes to the IgA plasma cell pool over time, and that these non-GC derived plasma cells can still be class switched, mutated, and positively selected which is in direct contrast to memory B cells, which class switch mutated memory does need to come from the gut germinal center. And of course, that all warrants a ton of further investigation, and we're very interested in doing that. But another observation I wanted to draw your attention to is this pattern of isotypes within gut inductive lymphoid tissues. So in the mesenteric lymph node and the Peyer's patch, we're looking here at flow cytometry data showing that most of the GCB cells within these two tissues are actually IgG1 positive. And if you think about the kind of function of the gut lymphoid tissues as generating these IgA secreted isotypes, this really doesn't make a lot of sense. And it makes even less sense when you look at the lamina propria plasma cell population, which is, to a rounding error, entirely IgA positive. So what are these IgG1 B cells doing here? Why are they in these gut germinal centers, and why do they make up the majority of them? So we wanted to do essentially the same experiment we did before, but in wild type mice, instead of the fate reporter mice, to investigate the relationships between IgA and IgG1 in gut germinal centers. This was done with the help of two of my lab mates, Shuting and Xiangyun, as well as, again, help from the Kleinstein lab, particularly Hailong, for this experiment. So the workflow is the same as before, and I won't go through it in detail. But again, we were able to get these same 3 basic clusters, which is really all we wanted from our gene expression analysis, and then turn over to our BCR analysis. So first, we wanted to look at the clonality of the IgA and IgG1 B cells. Our hypothesis here is that if these are representing 2 completely disparate responses, then they would not be present in the same clones at all. So we just made these 3 different logic gates, these 3 bins where we could put clones into. So clones can either fall into the yellow bin, which contain cells that have IgG1, but not any cells that have IgA. The blue bin containing cells that have IgA, but not IgG1. And then the green bin, which are clones that contain cells of both isotypes. And as you can see over here, when we look at clones with more than 10 members, we see a huge enrichment of clones that contain both isotypes. So that's kind of disproved our hypothesis that they're always separate. They do seem to be very interconnected in terms of belonging to the same clonal family and having a shared ancestry. I'd also like to point out, oh no, just kidding. But we are interested also to see how these B cells are related within a clone. You can be, part of a family lineage and be, related to somebody like Genghis Khan way in the past, or you can also have familial relationships with somebody much closer to you, like your parents or your siblings. And so we're interested in seeing kind of how closely these IgA and IgG1 B cells are related to each other, because that could have sort of more functional information in it as opposed to just being within the same clone. So to look at this, we made these big lineage trees, and these are a lot of these clones are very large, so the trees could get very complicated, and this is just a representative image of one, but I'll tell you about the patterns that we observed across multiple trees today. Here, what I just want you to pay attention to are the different isotypes. So this tree has mostly IgA and IgG1, A is in blue and G1 is in green. So we observed 3 main patterns in our trees and then quantified them. So firstly, we observed these IgA and IgG1 memory and plasma cells and GCB cells. It doesn't necessarily matter what their cell type is, but what we can see is that they're present at the same kind of horizontal distance from the germline on the same branch of the tree, indicating that they have the identical heavy chain mutation patterns. Now, just logically, it would be very unlikely for this to happen completely independently of each other, for these cells to generate these mutations in a way where they're not derived from a related common ancestor that's very recent. So we think that this means that these familial relationships between the A and the G1 B cells are very close. Similarly, we also observed this in 86% of the trees that we analyzed, which was an N of 56. We also observed these not exactly the same heavy chain mutation patterns, but similar mutation patterns where it appears that the IgG1 quote unquote sibling cell is in the germinal center and is highly mutated, whereas the IgA sibling cell is less highly mutated and is a plasma cell. We think this is occurring because once a cell differentiates and it leaves the germinal center and becomes a plasma cell, it's really not acquiring somatic mutations anymore, whereas this GCB cell is free to keep acquiring mutations and moving further to the right on our tree. So we think that this is another piece of evidence showing that these A and G1 B cells are very closely related at the time of sampling. And this, again, is very prevalent within the different trees we analyzed. It's not something that is cherry-picked out of the data. Now, to be kind of fair and objective, we know that the original isotype of naive B cells is IgM. So we wanted to look for IgM and IgA relationships between those two isotypes. And as you can see in this tree, there isn't IgM, but we did have IgM in other trees, although not super frequently. And we were able to find some of these relationships, but what we can see is that that's not really present in a ton of the trees that we analyzed. And the main criteria for analyzing these trees was size. We used trees that had over 100 members. So what could this potentially mean? Well, firstly, we don't think that IgM and IgA share a very recent common ancestor, at least at the time of sampling, which are antigen-specific B cells that we collected at 4 weeks post the initial immunization, but only about 5 days after the most recent immunization. But another hypothesis that we think could be explaining this is something called sequential class switching. So this was briefly introduced in the previous talk, talking about kind of reprogramming B cells that have already been class switched to switch to another isotype. And here what we're hypothesizing is that instead of a direct switch from IgM directly to IgA to make an OVA-specific IgA response, we think that there is a relevant, although perhaps not the only relevant, pathway where cells that have previously class switched to IgA or to IgG1 in the gut germinal centers are able to mature there and then class switch to IgA at a later time point. So we wanted to test this using a couple different metrics. Through our single-cell RNA-seq experiment, we were able to look at this a little bit. There was a recently published Nature Methods paper that developed a method for us to be able to look at sterile versus productive isotype germline transcripts. So a productive isotype transcript is something that's going to eventually make a BCR, but a sterile germline transcript is something that is transcribed from an intragenic promoter. So this is directly upstream of the isotype that the cell is about to switch to, and usually this transcription occurs when the cell is about to switch to a particular isotype or has already switched to that isotype. So this is kind of a marker of cells preparing to class switch. And what we can see are that these IgA germline transcripts, which are indicative of a preparation to class switch to IgA, are primarily coming from IgG1 B cells in the mesenteric lymph node and in the Peyer's patches. And then they're really not coming from any IgM or IgD positive B cells that we were able to pick up in our data. So perhaps IgM B cells were class switching to IgA at some point and contributed maybe to the early GC-independent plasma cells that we were seeing in the first half of the talk. But at least at the time point we're looking at, the active class switching that seems to be happening is primarily from this IgG1 ancestor. And this is something that we were able to verify through qPCR as well. So we're not just relying on a single single cell data set, but we're also looking at it ex vivo directly. Now, just because something is expressing a kind of germline transcript doesn't necessarily mean that that cell is going to class switch. So we also wanted to look at IgA plasma cells and see if they had IgG1 ancestry. So we did this by looking at this, the inclusion of this S-gamma1 region in the recombined switch region. And even though this assay is extremely insensitive, we were able to detect in distinct ex vivo samples that gDNA from IgA plasma cells does contain this S-gamma1 region, indicating that they have sort of a history of IgG1 switching. So to summarize the second part, we observed that IgA and IgG1 are very closely related in the gut and share these direct ancestor-descendant relationships, and that also we've shown that IgG B cells can sequentially class switch to IgA. Now, the last thing I want to leave you with is a question, which is why make IgA from IgG1? When I tell you that we have pondered this hypothesis and come up with so many different explanations over the years, it is definitely an understatement. But what we've settled on is that we think it makes evolutionary sense to have one B cell go through the whole rigmarole of somatic hypermutation, affinity maturation, et cetera, and then be able to then divide and differentiate into B cells of different isotypes that have different effector functions. So IgG1 is kind of an upstream isotype that is very good at providing systemic protection from pathogens. It has good relationships with Fc receptors, et cetera, and can circulate in the blood and prevent from severe disease once infected with something. But IgA is really essential at that mucosal barrier because it can cross the epithelium and be in spaces like the lung and the gut where IgG1 can't reach. So we think that this kind of simultaneous evolution of the IgG1 B cell allows it to then provide both mucosal and systemic protection through sequential class switching. Now, with that, I'd like to thank all of the many, many people that contributed to this work. If your name is on the slide, I'm very grateful for all of your help, and particularly for the Kleinstein lab, without whom none of the analysis would have been possible, because I started this project knowing nothing about the Immcantation pipeline or BCR sequencing at all. And I am happy to take any questions.
Thank you, Amelie. I'm afraid we're running out of time. We need to move to the next speaker.